and decided to try to make this thing, and it was incredibly hard to make. We worked on this back in 2021, right on the heels of the first X-ray structure of the SARS-CoV-2 enzyme in complex with a covalent adduct of Carmofur. Subsequently, after doing a bunch of, I guess, to just illustrate the, not really inefficiency, but the time burn and resource burn in wet lab projects, about 5,300 NMR experiments later, we have an optimized synthesis, which we published about 2022, Canadian Journal of Chemistry. We went on to make analogs in collaboration with Jerry Pazzi. And that came out earlier this year in Springer Nature Journal, Discovery Chem. And we have Chancey, Laverny, and Kathleen, who are here to share the next iteration of this. Actually, the third iteration of this product is currently in review in a journal, but because there's also a patent that Joey is part of that was covering part of that work, we can't share that. But the third part of this work, which we're not going to share today, actually uses a convolutional neural network deep learning algorithm to solve some of these challenges using image analysis. So Chancey, Laverny, and Kathleen are going to share a lot of what we've been doing in the wet lab. And without further ado.
Hi, I'm Laverny. I'm a rising sophomore at Santa Clara High School.
I'm Chancey, and I am a rising senior at Lindbergh High School.
And I'm Kathleen. I'm a rising sophomore as well, and I go to St. Francis High School.
So our project is the synthesis and evaluation of Carmofur analogs as membrane rupture inducing agents based on differing adjoint lipid tail lengths and varying substitutional isosteres. I'll go to the next. So here are the topics that we'll be covering today. An introduction and background of our project, analog design and synthesis, GUV rupturing kinetics and lipophilicity, as well as our future plans and acknowledgements.
So first, a little bit of background on our compound Carmofur. Carmofur is a prodrug of the clinically used chemotherapy agent 5-fluorouracil, and it is a really fascinating example of a repurposed drug that demonstrates dual activity as both an anti-cancer agent and also a COVID-19 antiviral agent. So in 1988, Carmofur was first discovered to have anti-cancer properties against colorectal cancer. And more specifically, it was discovered to be a covalent inhibitor of acid-ceramidase in 2013. And in 2020, amidst the COVID-19 pandemic, Carmofur was also found to be a SARS-CoV-2 Mpro inhibitor. So the way that Carmofur functions as an anti-cancer agent is by inhibiting acid-ceramidase. Acid-ceramidase is an enzyme that is responsible for the degradation of ceramide, which is an pro-apoptotic agent, which means that it's vital in carrying out normal programmed cell death. And when acid-ceramidase is overexpressed, ceramide levels drop and the cell avoids apoptosis. So this reduced apoptosis can then lead to more cancers, such as prostate cancer, where over 40% of tumors show elevated acid-ceramidase expression. However, by inhibiting acid-ceramidase, Carmofur is able to restore ceramide signaling, which then helps reinitiate apoptosis in cancer cells. Thus, it inhibits the growth of cancer tumors and cell proliferation. In addition, Carmofur, or the mechanism in which this happens is that Carmofur covalently modifies the catalytic cis-143 residue, which then deactivates the thiocarbamate and releases the 5-fluorouracil moiety. However, inhibiting acid-ceramidase is not the only function of Carmofur. More recently, Carmofur was also identified to be a potent inhibitor of the SARS-CoV-2 main protease enzyme, which is involved in the cleavage and maturation of functional proteins responsible for viral replication. So drug repurposing has actually been used for a really long time in order to optimize the speed of drug discovery, especially for new targets. So on the bottom left, you can see this chart showing that repurposed drugs usually take less time for regulatory approval. And this could be because it already has been tested for unexpected cytotoxic effects. On the right, it shows that repurposed drugs also have a higher percentage of success rate. So, drug repurposing is especially important for diseases that need to be urgently addressed, such as COVID-19. And aside from Carmofur, some other important drugs initially discovered as a result of drug repurposing efforts include zidovudine, which was originally used for cancer but repurposed for HIV, vingolimod, which was originally used for transplant rejection but then repurposed for multiple sclerosis, and topiramate, which was originally used for epilepsy, but then repurposed for obesity. So drug repurposing was what researchers turned to during COVID-19 because it was a means to develop a treatment really efficiently and with as much success as possible. So through high throughput drug repurposing screenings, Carmofur was identified as a SARS-CoV-2 main protease inhibitor. So this Mpro enzyme is involved in the cleavage and maturing of functional proteins responsible for viral replication. And its role in the replicase transcriptase complex is really crucial across different variations of COVID-19, which makes Mpro a really promising target for anti-COVID treatment. And the mechanism in which it inhibits the SARS-CoV-2 Mpro enzyme is really similar to the way that it inhibits acetoceramidase. So it covalently modifies the catalytic cis-145 residue and also deactivates the thiocarbamate and releases 5-fluorouracil. So the role of Carmofur in the inhibition of both acetoceramidase and Mpro, alongside other Carmofur analog structure-activity relationship studies, inspired us to design analogs that could enable more targeted and effective inhibition against acetoceramidase or Mpro.
The first issue we faced, however, was that the previous literature reported synthesis for Carmofur was really non-scalable. It was synthesized using pyridine, a toxic solvent, and the reaction required a temperature of up to 90 degrees and had a reaction time of 72 hours and still yielded less than 35%. Thus, for the first task for our earlier members was to optimize the synthesis route. They utilized benchtop 19F NMR, which provided real-time quantification of the relative reaction rates. Through this analytical method, they were able to identify a much more scalable reaction condition that utilized a greener solvent, NMP, and had a 90-minute reaction time, while still having greater than 70% yield. Their spectacular work was published in the Canadian Journal of Chemistry in 2023. The citation can be found on the bottom left corner, and there's also a QR code on the top right corner, so please check it out if you're interested. So now, with a successful optimization in hand, we turned our attention to synthesizing our Carmofur analogs utilizing this rapid reaction workflow.
So previous members of our lab designed a library of urethane and single-atom substituted analogs of Carmofur, and each group is designed to target different characteristics or properties of biological activity. And I would like to draw your attention to dodecyl and octadecyl urethane, as these analogs were designed to mimic the kind of lengthy lipophilic ceramides that bind to acetoceramidase. And as Kathleen mentioned, our results are published in Springer Nature in the QR code above.
Yeah, so with these compounds, we partnered with the Posi Lab to use GUVs, or giant unilamellar vesicles, to mimic the cell membranes and test their membrane rupturing potential. So in case you guys aren't familiar with what GUVs are, they're essentially cell-sized artificial membranes that can mimic the physical properties of biological membranes. They're generally used for drug discovery, studying membrane dynamics, as well as a lot of red blood cell research. And in our case, we're using them to test our compounds' membrane rupturing potential. So, using these GUVs, the PASI lab conducted a series of membrane rupturing assays. By taking before and after photos of the GUVs with our compounds at different concentrations and at different time points, they were able to quantify the percentage of GUVs ruptured by our analogs. So, if you look at the 4 panels on the slide, specifically the zoomed in images, after the addition of the octadecyl urethane, the GUV, which was originally very round and circular in panel C, began to look really patchy in panel D, signifying its rupture. So here's a bar plot of the average percent of GUVs ruptured after a 15-minute incubation with each of our compounds at different concentrations. So overall, you can see that on the bar plot, it indicates that our lipidated analogs of Carmofur, when compared to Carmofur, result in a much higher GUV bursting capability, with a decline in bursting activity starting at concentrations around 5 micromolar, but with some extension into the picomolar range. So also shown on the graph, we were able to identify the dodecyl urethane and octadecyl urethane as our hit compounds, as they have very potent membrane rupturing capabilities, with dodecyl urethane being able to rupture over 90% of GUVs, even in the micromolar range. So this really caused us to ask the question, number 1, what is the ideal lipid tail length for rupturing GUVs? How can we optimize this? And number 2, what is the impact of single atom replacement on the lipid tail? To probe this, we came up with a new library of analogs.
Building on what Kathleen mentioned in the previous discovery of our standout compounds, we decided to expand our library to various other substituents at the urethane moiety on Carmofur, here shown on the left. And through reactivity-guided design, we decided to probe the effects of dual inhibition of the main protease of SARS-CoV-2 and acid ceramidase, as mentioned earlier by Chansey, utilizing single atom substitution to form our carbamate and amide analogs, with the carbamate being in oxygen instead of the amine, and for amides in carbon. And this also allows us to see the effects of increasing the electrophilicity of these compounds and their metabolic stability. So to further explore the SAR, or known as structure-activity relationship, we decided to synthesize our steroidal and oleo analogs shown here on the left, which contain a saturated, which is the steroamide, and unsaturated lipid tail, which is the oleoamide. So this design was inspired by the previously reported oleoethanolamide by a group of researchers at UC Irvine in 2013, who discovered that this compound is able to inhibit acid ceramidase. Below are some of the other inhibitors that contain the urethane or carbamate moiety that are also acid ceramidase inhibitors. So here is our full library of compounds, including the varying alkyl chains that extending from 5 carbons, like 1.1 for our carbamates, and to 18 carbons, as shown as like 1.7 for our carbamates. And as you can see here, we also have some other interesting substituents that you may have not seen before, such as the 2-headed Carmofur on the bottom and the bis-cycle hexyl analog, as well as the cholesterol carbamate, to further attempt to improve the biological activity and potency of our compounds. Here on the slides, we have our carbamate series with the varying carbon chains and our cholesterol analog. And here on top, we also have our cyclopentyl substituted carbamate 1.2, which could potentially mimic the structure of a lipid tail, as well as possibly inducing a better fit in the binding pocket. Aside from that, our amide library consists of our saturated lipid tails, as well as our unsaturated analog, oleoamide, as mentioned previously. And lastly, our urethanes with the bis-cyclohexyl and 2-headed Carmofur, which could potentially be interesting to see what they target, and is quote-unquote fun and interesting by one of our group members. And to go over kind of how we synthesize these molecules, we have our starting material, which is 5-fluorouracil used as 1 equivalence, and we place it in a 2-neck round bottom with a Teflon stir bar. So attaching a septum to both of the necks, we filled the flask with nitrogen, utilizing the nitrogen balloon here as shown on the top right. And then we syringe in our NMP, which is the solvent, and syringe in our other reactants. It could be the chloroformate, isocyanate, or chloride, depending on which analog we're making. And then we add the base, which is triethylamine, and we use that to help speed up the reaction. And all of this is stirred at 60 degrees Celsius in a sand bath, and we are able to monitor the reaction through 19F NMR by seeing how the ppms shift over time. So here's an example on how, here's how like we ensure the purity of our compounds. So we're able to like see through proton NMR before we like send these compounds to bioassays. And here's a fully characterized NMR of our octaurethane analog with the amine on the 5-fluorouracil being the most deshielded proton, since it's next to 2 electron withdrawing groups, and the most upshield proton at around like 0.8 being the terminal alkene, which is a triplet at around 0.8 ppm. So as you can see, oleoamide was one of the compounds we initially sent to our high field, and our hearts really shattered when we received the oleoamide back from the high field, and we saw the peak at 12.0, which would tell us that our compound had decomped into 5-fluorouracil and oleic acid based on the carboxylic acid here shown at 12 ppm, and other characterization of the other protons.
So based on the NMR that Laverny just discussed, we decided to test exactly what is causing the decomposition of our amides. So we did suspect that the solvent DMSO was causing the decomposition, since all the amides that we sent through DMSO decomposed, whereas the heptoamide that we sent last year in chloroform D was completely pristine at high field. So in order to run these stability tests, we used our amide and we set up 2 NMR samples, one in DMSO and one in chloroform D. And as you can see above, the amide was completely stable in chloroform D, even after 99 hours and after we added water, and even when we left it at room temperature, it was completely stable. However, this was not the case with our DMSO samples. So from T0 to T96 hours, our sample appeared around 50% decomposed. So this means that as soon as we put our sample inside the DMSO, it started decomping. However, as soon as we added some more water, the decomp visibly increased. And within 2 hours after the addition of water, the sample was fully decomposed. So through this, we know that a combination of DMSO and also water is making our amides decompose. And as a result of this test, we are deciding not to proceed with the amides in biological assays as they are simply not stable.
So now with our remaining library of analogs, the Palsy lab decided to test our new compounds in GUVs. So in membrane-rupturing assays, the dodecyl urethane did very well, being able to consistently pop all the GUVs in all concentrations down to 100 attomolar. When the amine on the tail is instead switched to an oxygen, as in the case of the C12 carbamate, the compound began to show decreased activity, but was still able to consistently pop over 90% of GUVs down to the picomolar range. Similarly, the octyl urethane also did very well, being able to pop 60 to 90% of GUVs even in the femtomolar range. And to kind of visualize just how dilute femtomolar dilution is and just how potent our compounds are, if 1 mg of our product was the size of an ant, you would need to drop it into 1,000 Olympic-sized swimming pools to make a femtomolar dilution. And our compound at this concentration is able to pop all the GUVs in under 2 minutes, making it one of the most potent membrane-rupturing molecules in science. So to gain more insight into the physical properties of the compounds, the POSI lab is currently attempting to identify the critical micelle concentration, or the concentration at which our compounds begin to form micelles. So in case you don't know what micelles are, they're spherical or cylindrical aggregates of surfactant molecules. They're formed when amphiphilic molecules, which have both a hydrophobic and hydrophilic part, are dissolved in a liquid, and the hydrophobic tails would cluster together as shown in the figure, and it would form the core of the micelle, while the polar hydrophilic heads form the outer layer and is in contact with the surrounding liquid. So to calculate the CMC, as you can see in the graphs, the POSI lab is measuring the contact angles between the GUVs and the slides. So when you see a sharp change in the contact angle, you can, as you can see in the 3 graphs, you would find the point of inflection, or like estimate it, to find the CMC.
So our future work involves investigating the optimal carbon tail length for the maximum efficiency for GUV membrane rupturing. And additionally, we need to analyze the CMC data for a connection between self-assembling ability and membrane rupture capabilities with our compounds. Lastly, we hope to test our compounds in vitro through MTT and LDH assays to determine if there's a correlation between GUV membrane rupturing abilities and cancer cell membrane permeability.
So we would like to thank our other Carmofur project members for all their support on this project, as well as our lab alumni who have been working on this project for many years. In addition, we have collaborators in Dr. Pazzi's lab, and we'd also like to thank our advisor, Dr. Edward New, as well as Steve Lynch, who helps us run our high field NMRs at Stanford University. And if you're interested in reading more about our project, we have currently 2 publications. The QR codes are on the bottom right. And thank you for listening.
Oh. All right. Questions?